Hello, everyone, and welcome to today's webinar, Data Safety Monitoring Boards for Adaptive Trials. Before we get started, I would like to go over a few items so you know how to participate in today's event. You have joined the presentation listening using your computer's speaker system by default. If you prefer to join over the telephone, just select telephone in the audio pane and the dial-in information will be displayed. You will have the opportunity to submit text questions to today's presenters by typing your questions into the questions pane of the control panel. You may send in your questions at any time during the presentation. We will collect these and address them during the Q&A session at the end of today's presentation. Today's webinar is being recorded and you will receive a follow-up email within 24 to 48 hours with a link to view the recording. I would now like to introduce our presenters. Scott Berry, PhD, is President and Senior Statistical Scientist at Berry Consultants, LLC, where he works on the design of innovative clinical trials, Bayesian statistics, and clinical trial simulation. He earned his PhD in statistics from Carnegie Mellon University and was an assistant professor at Texas A&M University before co-founding Berry Consultants in 2000. He is adjunct faculty in the Department of Biostatistics at the University of Kansas Medical Center. Dr. Berry was elected as a fellow of the American Statistical Association in 2013. I would now like to introduce Michelle. Michelle Detry, PhD, is the Director of Adaptive Trial Execution and a Senior Statistical Scientist for Berry Consultants. Dr. Detry is a biostatistician with expertise in the implementation of adaptive clinical trials, including platform trials. As part of her role in implementing Berry Consultants' trial designs, she is a member of numerous statistical analysis committees that conduct the interim analysis. In addition, she has expertise in clinical trial design, reporting for data and safety monitoring boards, and also serves on DSMBs. I will now hand over to Scott.
Appreciate it. Thank you, Anisha, and appreciate the invite. What I thought we'd do is I will start and I'll talk about some adaptive trials, give some examples, just very quick run through some example trials, and then I'll turn it over to Michelle. At Berry Consultants, Michelle leads our efforts in implementing trials, frequently interacting with DSMBs. And it's a role actually within adaptive trials that I took a little bit for granted as we started, and it turns out to be really a critical thing in how they interact in their role within adaptive trials. So I'll try to set up what adaptive trials look like, and then Michelle will talk more about their role in the trials. All right. Okay, so I'm going to go through a couple examples and then turn it over to Michelle, and then we should leave ample time for discussion. So please, if you have questions, put them on there, and we'll hopefully have a lot of time to discuss those. All right. So my first example is the Trulicity trial. Trulicity is the drug name, and I'll describe a little bit of this trial. And again, my goal is to see a little bit more complex, innovative designs, the complexity of them, the role of simulation, the role of modeling, the role of models and thresholds and pre-specifications in these trials, and hence then what are the roles of DSMBs within them. So Trulicity is now an approved drug in diabetes. As an experimental drug, the phase 2 trial for Trulicity was a phase 2/3 seamless trial. It had 7 active doses of Trulicity, a placebo and an active control. The trial enrolled 50 patients across these 9 arms, It was approximately 6 per arm. And then adaptive randomization started. So there was a burn-in period in the trial, and adaptive randomization started at 50 patients. The figure doesn't show the interim starting at like 54 patients. And then every 2 weeks during the trial, interim analyses were conducted. The interim analyses were for response adaptive randomization, so it kept the rate on placebo and active comparator constant, but it increased the rate over the doses that were working better in the course of the trial. Was based on 4 endpoints: HbA1c change, which is the primary efficacy endpoint. Weight loss was a measure, and I'll describe how they all went in very briefly. Blood pressure and heart rate were safety endpoints in the trial. They were all built into a utility function, and we increased the rate for arms that were doing better on this utility function, decreased the rate for others. When 200 patients had been enrolled in the trial, the first interim in which 200 had been enrolled, again, these were on Wednesdays, every other Wednesday during the course of the trial, there were triggers that could trigger going to phase 3. The algorithm would select a dose or 2 based on predefined rules, and if it shifted to phase 3, it would do 1-to-1 randomization between all arms at that point in the trial. The sample size was selected by the algorithm at the time of transition, and it could transition anytime from 200 up to 400 at these fortnightly interim analyses. The modeling within the trial was all prospectively defined, and I will move this slide forward. There we go. So there were 4 repeated measures models over those 4 endpoints. They were combined together in a single utility function. The Bayesian algorithms were calculating the predictive probability that if the algorithm shifted, it would be successful in the primary endpoint. At the end of the trial, the final analysis would incorporate all of the doses that continued forward, and it would include patients that were enrolled during the more learning phase 2-ish phase of the trial. We pre-specified a weight function for these. Each component of the endpoints were weighted in this predefined algorithm. The better HbA1c control, the higher the utility for the dose. The more weight loss, the higher the utility for the dose. But if pulse rate and blood pressure were increasing, it would decrease the value of the dose. And if the increase was great enough, it would say that dose has 0 value because it likely has cardiovascular risk issues. That algorithm was run every Wednesday during the course of the trial using all the updated data on those endpoints. We simulated this trial. We demonstrated control of type 1 error using an ANCOVA model at the end of the trial. So it was a frequentist analysis at the end, even though a Bayesian analysis was driving the adaptations. You're welcome to read the paper about some of the details of the trial. When the actual trial ran, after we had run it 33 million times in silico, it got to 200. The interim analyses were conducted. It got to 200 and said, you need to go to phase 3. And it selected 2 doses, the 0.75 and the 1.5 milligram doses were selected to go to phase 3. When that selection was made by algorithm, it triggered other phase 3 trials, a cardiovascular risk phase 3 trial, and then other phase 3 trials for efficacy in the development program on the doses that were selected. Trulicity ended up being very successful in the phase 3 component. The cardiovascular risk trials were quite successful. And in 2014, the US FDA approved Trulicity, and it's now in the US, it's a $3 billion a year drug. And Eli Lilly says that trial, that phase 3 trial, saved them anywhere from 6 to 18 months of development time on patent life. in the acceleration of the 2-3 trial. But you can see how pre-specified and how algorithm-driven that trial was. Another trial similarly pre-specified was the DAWN trial. This was a device trial, thrombectomy for those suffering acute stroke. Individuals would come in, and these devices were approved at the time up through 8 hours. So if time lasts seen well, subjects within 8 hours. So this trial was to look to see if subjects that are longer than that time period, 8 to 24 hours, would also benefit by these clot clearing devices. They were worried that in some patients, this device would not be beneficial. It might even be harmful. Those that have difference in the size of the infarct in the clinical outcomes, they thought would benefit the most. So they had a prospectively defined enrichment that during the course of the trial, they could stop enrolling the larger infarct size to the smaller ones with the larger clinical mismatch. We designed an algorithm that would do predictive probabilities over the type of patient being enrolled. Every 50 patients during the course of the trial could enrich and in defined 5 milliliter chunks in the infarct size, or it could stop for futility. We adjusted for the potential subset analyses, the cherry picking in the trial and a final analysis. The trial could stop for expected success if the current patient population, if all followed through the 90-day outcome, I can't remember, 60, 90-day modified Rankin score, if that would then be successful, they stopped enrolling, and then the final analysis happened when everybody got through the final time period. Or the trial could stop for futility if enriching to the smaller stroke sizes was not beneficial. And this was all entirely by algorithm. A robot could run these interims and make decisions in the trial. Michelle will talk a little bit more about what those robots look like and are humans watching the robots and how does that all work. The trial got to the first time where it got to 150 and it did not enrich, but it couldn't stop at that point for success, and it didn't stop for futility. So I was blinded to the results. Others were conducting interims, and we found out no futility, keep going. And then when it got to the 200 patient interim analysis, it said you need to stop enrolling. The trial's very likely to be successful with no enrichment with that sample size. They were followed for the full 90 days to the 90-day modified Rankin score, and then the final analysis was conducted. The final analysis is a Bayesian analysis of the ordinal outcome, and you can read the paper on this. It was incredibly successful. The device works incredibly well, huge benefits, and the US FDA approved this now out to 24 hours in this patient population in great part to this particular trial. Now, the adaptations didn't necessarily happen, though it stopped at 200 patients rather than the maximum sample size of 500 in the course of it. So that's, again, a rather complicated simulation-based trial. By the way, here's a good example if you're interested in Bayesian stuff being reported in medical journals. All of this is posterior probability of superiority and the Bayesian primary analysis in the trial. And I'll just very briefly say something about Precision Promise. So Precision Promise is a platform trial in pancreatic cancer. Its sponsor is a patient organization here in the US, probably global patient organization, PanCan, and they're sponsoring the trial. There are a lot of companies that are avoiding pancreatic cancer because there aren't any successes in the treatment of it. The diagnosis is grim, and so they're trying to get more and more drugs, more and more shots on goal in pancreatic cancer. So it's a platform trial where many drugs will come in. They will share the infrastructure. they will compare to a common control arm. So these are one of the movies we put together, and I'm not going to go through this, just to show that during the course of the trial, every month, randomization probabilities are set across all the arms that are in the trial. Decisions are made about each arm. Should it advance? Should it stop? Should it continue only in a subset? And all of this is done entirely by algorithm. So this is a dashboard of what you might see while the trial's going on the individual decisions in the trial, which is, again, pre-specified algorithms, thresholds that could be run by machine with, of course, potentially a DSMB and a statistical analysis committee watching it. I won't say much about the complexity of the trial. The next slide goes through many of the adaptations and the complexity of what happens every single month with the patient download, randomization probabilities reset, decisions reported on all the arms in the trial during the course of the trial, and this is done monthly in these trials. So I wanted to give you a flavor of the complexity of some of these trials, and then I described this idea that this could be automatic pilot. Well, it may be automatic pilot, but we have people watching, and now Michelle is going to describe who's watching these algorithms and the role of the different committees. Michelle?
Okay, great. Thanks, Scott. Anisha, can you hear me? Or Scott, can you?
Yeah, I can hear you.
Thank you.
All good.
Okay, so thanks, Scott. So tagging along to those designs, wanted to talk a little bit then, of course, about the conduct of them and how a DSMB interacts with those type of designs. A DSMB still has its usual role. Nothing changes as far as their role of protecting human subjects, ensuring the proper conduct of the trial, the ongoing scientific validity, integrity, and clinical and scientific relevance in the study. And they still review their standard unblinded DSMB reports with the safety summaries and all the details that are prepared by an independent statistician or independent statistical team. Just getting my controls here. Bear with me as we wait for the controls to switch. Well, I will keep talking while I'm working on getting the mouse control to switch. So I talked about how they have the usual role, but in addition to this role, the DSMB now has to monitor the conduct of the trial, which includes the adaptations. And these adaptations should be implemented as specified in the protocol. You heard Scott say several times, the algorithm is pre-specified. You're going to hear me say that a lot of times too. And that's really a key here, and that's an important thing for the DSMB to understand. The DSMB must be unblinded to treatment assignment. DSMBs, regardless of whether it's an adaptive design or non-adaptive design, should be unblinded to treatment assignment so that they can assess the risk-benefit considerations. Sometimes we do unfortunately see that people think DSMBs should be blinded. That is not the case, and especially is not the case in adaptive trials. And also the DSMB has to be allowed to review all trial data, including outcome data as an endpoint. Sometimes because it's a data safety monitoring board, people think that DSMBs shouldn't be allowed to see outcome data, but they need to. It's the only way they can assess the risk versus benefit, and especially essential in the adaptive designs. All right, great. So additional considerations. So since this DSMB has now these additional responsibilities, it's crucial that they understand the design and what the trial is supposed to do. And the reason they need to do that is because they have to be able to assess if something's going wrong, and this will be with the help of a statistical analysis committee, and I'll talk more about that group later. They also need to assess if the trial's pre-specified actions are no longer appropriate, and they need to recognize the potential negative impact on trial validity of making design change recommendations after they've seen unblinded data. Next slide. I think, Anisha, you may have control, so I can just say next slide and you can forward. Next slide. So in order for the DSMB to understand the design, the kickoff meeting is a key activity. And this should hopefully occur before enrollment begins. Sometimes just due to timing, that's not possible, but it should occur very early on, at minimum when the trial starts. A kickoff meeting contains the usual DSMB training, but then the DSMB also has to be trained on the adaptive design. And what that includes in a kickoff meeting is probably a little more time than other kickoff meetings for the design team to describe the design and to show example trials. And Scott showed you the Precision Promise, that big dashboard, and that's part of an example trial where we actually show situations where we simulated patient data and then we run it through the trials and we show the DSMB members at each interim analysis what happens based upon the data that's being viewed so they understand how the trial should perform. Next slide.
Michelle, I think you should have mouse control now. If
I
you can
do.
try
Thank
and
you,
move your
Anita.
mouse around.
I've got it. Thank you.
No worries.
So as we hit the interim analyses, we talked about how the DSMB is responsible for monitoring and making sure the interim analyses are run as according to the protocol. And how they do this is they are going to review interim analysis reports. These are prepared by the Statistical Analysis Committee. They're provided to the DSMB for informational purposes, and they're supplemental to the usual full DSMB report. They're a small report. They're going to specifically have the interim analysis results. They don't have any safety information. They just have the outcomes used in the interim analysis and then some subject disposition data to understand enrollment numbers, how far patients are along in the trial, lost to follow-up, things like that. As I mentioned, we're going to say this many times, the interim analyses are pre-specified, so DSMB deliberations when they're reviewing interim analysis reports must be guided by the trial design because it's very, very important to follow those rules and those algorithms that Scott already showed. So the key here is that the trial may be adaptive, but the DSMB is not adaptive. This is not a situation where the DSMB looks at the data and decides what to do. The algorithms and the corresponding adaptations that should be conducted are specified in the protocol and should be run as planned. So the reason I bring that up is there's sometimes been situations where DSMB members that are new to adaptive trials may consider these adaptations, for example, stopping boundaries, stopping for expected success, like you saw in the DAWN trial, stopping for futility. They may view them as guidelines, not rules. But if they view them as guidelines and decide to deviate from the design, there are consequences. The design is no longer pre-specified, which is a top criteria of adaptive designs. When it's no longer pre-specified, the trial operating characteristics such as type 1 error, false positive rates, and trial power are no longer known. The decision to deviate from the design likely introduces bias because the decision was made with knowledge of unblinded data, and that bias is unable to be quantified, and therefore when the trial completes, the trial integrity may be questioned. So, of course, the DSMB can always make recommendations to deviate from the design or what the design says to do based upon patient safety. So this adaptive design in no way hamstrings or restricts the DSMB from the usual role in monitoring patient safety. It's really these non-safety recommendations to deviate that could be problematic because really they're technically protocol deviations and they can have the consequences we just discussed. So one of the things I like to do in DSMB charters is put in language that a DSMB can talk to regulatory authorities before recommending deviations. And I quoted here below a little snippet from the FDA adaptive design guidance that when talking about the importance of pre-specification adaptive designs and how that pre-specified adaptation should be followed, it says something, it says the following: when completely unforeseen circumstances arise, we recommend discussing any potential design changes with the FDA as soon as possible. So the reason I bring this up is if the DSMB finds themselves in a situation where they really need to deviate from the design, there may be a reason why. And this is a trial that's going for confirmatory, for regulatory approval, for maybe confirmatory for approval of the drug. And they may need to or want to talk to the FDA to make sure what they're recommending doesn't jeopardize the ability of that trial to be used for the approval that it was intended. So that's something I like to put in the charters to allow that to happen ahead of time and not be something the DSMB has to ask to do, because that would be a kind of a providing information to blinded trial team members when they ask. So this should be something that is in place ahead of time. DSMB meeting timing. Do the DSMBs need immediate interim analysis? We get this question a lot. And my answer is it depends. And I've got a couple examples here of situations. Scott gave you an example of the Trulicity trial, which had the response adaptive randomization applied every 2 weeks. In this situation, the updates to the randomization probabilities, they're basically part of the protocol. The DSMB does not have to look at those results each time and approve the updates to the randomization probabilities. It's the same sort of thing as in a randomized one-to-one trial that the DSMB doesn't have to approve those randomizations each time. It's part of the protocol. However, the DSMB needs to be kept aware of these randomization probabilities and how they're changing over time, because they're monitoring in the background the safety also, so they need to know how they're changing in case they were watching a potential safety signal. So they still need to know what's going on, but they don't have to officially approve each one and have that be part of the process. However, a DSMB may want to have a meeting and an interim analysis where a major stopping decision is triggered. For example, where Scott talked about in the Trulicity trial again, where it reached a criteria where the subsequent trials were spawned, or the Dawn trial where the threshold was reached where the enrollment was to be stopped for expected success. With these major stopping decisions, it may require a DSMB review before these actions are transmitted to the team, because it allows the DSMB just to do a review at the safety, make sure they're comfortable with the decisions in the safety profile, and to add on any safety recommendations if desired. Now, you can see from the trials that Scott showed that when we do these analyses, when the analyses happen, they're pre-specified, and the DSMB has to be ready to review, meet, and react quickly to the results. So DSMB members have to be nimble. Sometimes DSMBs want to have 2 weeks to review materials, or they sometimes have to have their meetings scheduled 3 months in advance. That doesn't really work well with an adaptive trial, because we can't have those kind of delays after an interim analysis is conducted before the interim analysis result is conveyed to the trial team and the action implemented. So DSMBs for adaptive trials need to understand they need to be nimble and may need to react quicker, review quicker, turn around decisions quicker, and make themselves available. So following up on that, since the DSMB members have to be nimble, that needs to be considered when you're selecting who's going to serve on your committee. Key opinion leaders often are very overscheduled and may have limited time. And so this should be discussed when you're considering your members to make sure that they can react quickly and review things quickly and have meetings with more limited notice. So not saying that key opinion leaders are not good DSMB members, they just need to understand the demands of the design and what they need to be able to do. Another criteria for DSMB member selection is experience in adaptive designs. You want to have some to most of your DSMB members have served on DSMBs for adaptive designs before. If you have an entire DSMB that's new to adaptive designs, that could be problematic as far as them understanding their role. The DSMB should, of course, have no preformed opinions regarding the intervention, as with all DSMBs, but they also should have no preformed opinions regarding the design. When they sign on to be part of the DSMB, they're signing on to make sure the design is implemented as planned, and that they are agreeing with that design plan. And then you need to balance necessary expertise with conflict of interest requirements. And I like this slide that Roger Lewis often uses that shows how there's usually a dichotomy here with the expertise and the conflict of interest, where those DSMB members may have the greatest expertise, may also have the greatest conflict of interest. So you need to be careful and considerate of what conflict of interest requirements you have to not make it so strict that you don't get any experienced DSMB members, but also make sure you have the appropriate conflict of interest for the independence of your board. Okay, that Statistical Analysis Committee we kept talking about, the SAC. Sometimes it's also called the Statistical Monitoring Committee, an SMC, or an ISAC, Independent Statistical Analysis Committee. There's several different names that it goes by depending on the trial. But this is supplemental, separate from the DSMB. It's a small unblinded team, and it's made up completely of statisticians. It may have 2 to 4 statisticians. The size really depends on the type of trial and the number of adaptations and the frequency of adaptations. So for example, a platform trial like the Precision Promise trial that Scott was talking about is going to have a larger team because it's going to have monthly analyses and more moving pieces. More smaller, straightforward trials may have a smaller team of just 2 statisticians. These statisticians need to be experienced in adaptive design and the relevant statistical methodology. So for example, in the DAWN trial, where it was a Bayesian design, the statisticians need to be experienced in Bayesian statistical methods and understand the Bayesian models. And I highly recommend that this committee include at least 1 member that was on the design team. Scott mentioned how on the DAWN trial that he was blinded and there were other members from the design team that were on the statistical analysis committee that were unblinded. And so what we did was Scott was on the blinded side working with the sponsor, answering questions about the design. But then another team member that worked with him on design was on the SAC and making sure that the interim analyses were performed as planned. And the reason that's important is that statistician has the institutional memory of how the design came to be and why decisions were made and why adaptations were selected to occur where they did and what the desired actions were. And that's important in case some weird situation happens where decisions have to be made as far as what the algorithm should do. Pre-specification is key, but I think we all know that sometimes there may be a little weird data element that happens that we have to decide what's best to do, and we want to make sure we want to do it in the spirit of how the trial was designed. The role of the SAC, if they're not voting members of the DSMB, they're not part of the DSMB, what they do is they perform the interim analysis. They're kind of the robot Scott was talking about. So they're performing the interim analysis, specifying the protocol, but yet they have that human element to make sure they're doing what it's supposed to do. And doing what it's supposed to do could be, making some decisions based upon what they're seeing with the data. But the main goal and what usually happens is it goes exactly as specified in the protocol. They're not doing any secondary or safety analyses. The team is receiving limited data, only the data directly used in the interim analyses, and subject disposition, number of subjects enrolled, how far along they are in follow-up. They're not getting safety data. They're not doing all the other summaries that the usual full DSMB report has that the other team supporting the DSMB is doing. This interim analysis report is created, it's supplemental to the full report, and they interact with the DSMB, and then they sometimes have a separate charter. The situations where they most commonly have a separate charter is in a platform trial or maybe a more complicated trial where there's a little more description that's needed as far as the role. And the reason we have a separate charter is sometimes there are details in there that are really specific to them and would kind of be extraneous information that's really irrelevant for the DSMB charter. But of course, the DSMB has access to the charter to understand the role and responsibilities outlined for the SAC. So a diagram here. So how all this works. So let's say, for example, you have all your sites that are enrolling the patients, and they're interacting with, of course, the randomization system, drug supply, if this is a drug intervention trial, and then the database where they're entering data, your EDC. And then we just talked about these 3 groups. We've got the DSMB, we've got the independent statistician or independent statistical team supporting the DSMB, and then the statistical analysis committee. The randomization treatment assignments go to both the independent statistician supporting the DSMB and the SAC when they have data extractions. Data from the database goes to both groups. And then both groups are preparing their respective reports for the respective roles and interacting back and forth with the DSMB. The independent statistician supporting the DSMB is doing the full safety usual DSMB reports you may be familiar with. And the SAC is doing the interim analysis reports at the time of the interim analyses. The SAC may have an additional role if there's response adaptive randomization, like in Trulicity or Precision Promise, where they are transmitting randomization probabilities back to the randomization system. And then the DSMB is providing communication to the sponsor representative as far as any safety recommendations from the usual role and what the interim analyses say they should do. These three groups, the independent statistician supporting the DSMB and the SAC along with the DSMB, reside within a sphere of confidentiality. These are the unblinded groups, and this sphere of information needs to be carefully controlled so that the highly confidential information they have about what's happening in the trial is not leaked outside of that sphere. So a little further along that, restricting the access to the interim results. Whenever we do an adaptive design, and whenever the adaptive designs are reviewed for regulatory purposes or in the scientific literature, there always is a concern about the potential for operational bias. And operational bias results when information from the ongoing trial causes changes to participants' enrolled, investigator behavior, or other clinical aspects in such a way that the conclusions are biased. And this is a key concern for innovative adaptive trials. Almost every regulatory guidance document you will see from regulatory authorities will talk about the need to limit and control the possibility of operational bias. And for example, the previous FDA draft guidance had a whole specific section and big highlight on it. And it's definitely included and discussed in the current FDA guidance on adaptive trial designs, and also in the EMA guidances too. And to do this, it requires planning and documentation. So we just saw the three groups are in the sphere of confidentiality, and it requires plenty of planning of who they communicate with and documented plans for how that'll work. So that ties into a data access plan, which is really kind of known as who knows what when. This is referenced in the FDA guidance for adaptive trials. And basically it includes who are the unblinded personnel, how is access controlled, are there firewalls between groups? For example, Scott was talking about how he was blinded on the Dawn trial, and so it would describe the firewalls between him and the SAC. It should talk about how adaptive decisions will be made. And that's pretty straightforward because it should be made as pre-specified in the protocol. It should talk about the type of information disseminated and to whom. And a lot of these details may be partially in the protocol, maybe in the statistical analysis plan, and a lot of it's often in the DSMB and SAC charters. And this is a document that has to be frequently submitted to regulatory bodies during the design stage. Just trying to move forward. Okay, so we talked about, you saw in my picture that we have the sponsor communication contact. And so the reason it's important to have this contact defined ahead of time for the DSMB, because they're going to receive not only DSMB recommendations on safety, but also the high-level result of the interim analysis. This sponsor communication contact needs to be separate from the study team, which means they're not involved in day-to-day trial activities. And the reason of that separation is they have to receive limited unblinding information because they're the one receiving the resulting action from the interim analysis and any sort of safety recommendations from the DSMB. This contact needs to have the seniority to make decisions and act on these communications and recommendations. And then eventually they'll transmit any resulting actions from the adaptive design to the study team. So this becomes part of the DSMB communication plan, which needs to be clearly outlined in the DSMB charter. When the DSMB communicates results of interim analyses, only the high-level results are transmitted to the sponsor contact. The results such as continue the trial, stop for futility, stop for expected success, for example. They don't get the probabilities that came with these decisions. They just get the decisions that were triggered based upon the resulting underlying analyses. And then there are two models that we see for how these interim analysis results, high-level results, are transmitted. Sometimes, and most often, they're sent to the DSMB, and then the DSMB communicates it to the sponsor contact. Sometimes they're sent directly from the SAC to the sponsor contact with a copy to the DSMB. A lot of this depends on the type of design and the situation, so there's a lot of it depends here. But the procedure is specifically outlined and detailed in the DSMB charter and the SAC charter if it applies to the SAC. But regardless of which model is used, the DSMB always sees all the interim analysis reports, regardless of the communication plan. A DSMB always needs to know the details of what's happening in these interim analyses so that they can monitor the trial and fulfill their role. They should not be restricted from seeing these results. The resulting DSMB communication letters, again, they only have these high-level results, but these communication letters are really an art because the recommendations need to be carefully constructed to contain the minimal amount of information necessary. The DSMB is in that sphere of confidentiality and knows a lot of information, and they need to be careful about what they say and what they reveal. So they want to contain minimal amount of information in their letters, and specifically, if they have a recommendation, they don't always have to provide the rationale. Rationale for a recommendation may or may not be given in whatever way best maintains the scientific integrity of the trial and minimizes the leakage of unblinding information. So the SAC interacts with the DSMB by participating in the closed sessions of DSMB meetings where unblinded results are discussed. This will usually only happen at interim analysis time points. DSMB meetings may occur before the interim analyses begin. The SAC does not need to be part of those unless the DMC requests they be there. And the reason they participate in the closed sessions when interim analyses are conducted is so that they can explain the results and answer questions. They also respond specifically to DSMB requests for additional analyses if necessary for the DSMB to understand what's going on. And then if the DSMB wants to recommend to deviate from a pre-specified design, this should be discussed with the SAC to understand implications. And so it's probably, I haven't given a lot of detail on what kind of rare situations could happen where a DSMB may want to deviate from the pre-specified design. And these should be very rare and unusual situations. One could be, let's say, something seems to be off with the randomization probabilities. Maybe the response adaptive randomization didn't get turned on. There may be some sort of decision that was triggered that shouldn't happen. Another situation could be the quality of the data. Maybe there's an outstanding amount of missing data where the design, some sort of adaptive decision is triggered, and it's really concerning that that's the correct action to follow because there was so much missing data. So this isn't a situation where the DSMB is looking at the data and deciding what to do. They're really deciding whether the protocol is being followed as designed. And so if they have one of these situations, they need to work with the SAC, discuss it with them, to understand not only what is going on in the data and the analysis that was conducted, but to understand the implications of them deviating from the design. And this whole process and this whole interaction should be outlined in the DSMB charter and discussed in the kickoff meeting. A few logistical considerations just to wrap up. We talked about whether the interim analyses will coincide with the DSMB meetings. Another logistical consideration is whether there needs to be a full DSMB safety report with each interim analysis report. You may have seen that these full DSMB safety reports are quite large and long and take a while to produce and also a while to review. You may not need to have a full report with coincide with each interim analysis report. If it's just for updating the randomization probabilities, again, the DSMB doesn't have to review and approve. The DSMB might just have their meetings scheduled as they were originally intended, and they will be kind of close enough that they had a recent safety review. A DSMB may also decide that they want to have a small abbreviated safety report with certain outcomes at the time of interim analyses so that they can monitor certain safety signals of interest. And this should all need to be worked out during the kickoff meeting. And then also, it's really important to think about aligning the data cuts for the interim analysis reports and the full DSMB reports. If a DSMB is reviewing two reports at the same time, they need to know that this comes from the same data set. And if it doesn't, they need to understand that ahead of time and what the difference is between the two sets. I bring this point up because the interim analysis reports are going to be much faster to create and to conduct the analyses because it's smaller than a full DSMB report. So they're turned around pretty quickly. In a more straightforward trial, and most of our trials, we try to turn around our interim analysis reports within 5 business days. It may go up to 10 business days, depending on the number of analyses or what all needs to be happen. But we usually try to keep our interim analyses pretty reduced so that we can perform them quickly because randomization is continuing in the background. And we want to make sure there's not a lag in the decisions being implemented from when the interim analysis began. So it's really important to be able to understand what the DSMB is receiving as far as the the data that's in their reports and try to align them so they're seeing the same patient populations. And then my last slide is just a summary slide. There it goes. And so in adaptive design, DSMB, they require additional considerations, including member expertise, availability, and training. The analyses need to be conducted as specified in the protocol, and the DSMBs need to understand the trial integrity consequences of not following the pre-specified design. This is not meant to keep them from making safety recommendations whatsoever, but it's just the importance of the pre-specified design. And then the key concern always of maintaining confidentiality of these comparative interim results and preserving trial integrity and making sure information is not leaked. And with that, Scott and I can take questions.
Thank you, Michelle and Scott. We are now going to begin answering the questions submitted during today's presentation. As a reminder, you can still submit your questions through the question pane in the attendee control panel. If you would like to address a specific presenter, please make sure you include their name before the question. I will now start reading out the questions that have been posted already. And the first question is from Ian Marshna. And Ian is asking, I'll just give this one to you, Scott. In Trulicity, including data from RAR phase in the final analysis introduces some conditioning. Does this need to be accounted for?
So thanks, Ian. So it introduces some conditioning. So by including the phase 2 data, we started with 7 doses, and the 2 of them were selected to move forward in the trial. Now, there's a cherry picking aspect of that good doses are moved forward. It's slightly more complicated than that because it was a weighting of safety and tolerability and efficacy. So it's not always the single best, but you can get a cherry picking from the early part. So in order to understand the penalty of that cherry picking, we did simulations. And we interacted with the FDA on this, and they asked, okay, we'll take the simulations, but we want 70% of the data to come from the phase 3 portion. And given the indication and given the space, that Lilly was completely fine with that. So I don't know if you noticed, but the ANCOVA analysis had a 0.020 rather than a 0.025. So we did many, many interim analyses, did the selection, and the penalty, if you will, was to go down to 0.020. The EMA, in reviewing the trial, said, and it was Rob Hemmings, a statistician, who said, we've run out of reasons to say no to that analysis, so they accepted it. So it was one of the very first simulation control of potential increase in type 1 error. But we did account for it, yes.
Right. Thanks for that, Scott. Our next question is from Stefan Herishier. Would you like to take this, Michelle? The question is, how do you train DSMB members?
Oh, sure. So I like to do the training of the DSMB members in the kickoff meeting and making sure that we have a long enough meeting to describe to them the adaptive design, the need to follow the pre-specified design, make sure they understand the design, allow them plenty of times to ask questions, and then to go through the charter in detail, because I include a lot of language in the charter regarding a detailed language in the charter regarding the roles. I like to provide that charter to them ahead of time so they have time to review it before the meeting so that they can come prepared to ask questions. And as far as training the DSMB on the design, we really feel that showing adaptive trials are the best way to help showing example trials, excuse me, are the best way to help the DSMB members understand the design and the complexities. So we spend a lot of time simulating trials like Scott just talked about to get the operating characteristics, but then we take individual trials that we simulated and we create graphics as far as what the data is showing at each interim analysis, and then what the resulting decisions were. And we can walk the DSMB through what happens at each interim analysis. And we find that extremely useful in training the DSMB on the design. And at the same time, we can talk about the need to follow this pre-specified design and potential consequences if they don't follow, what should happen according to protocol.
Can I jump into that, Michelle? So since I showed the Trulicity trial, the DSMB for that trial was told they couldn't change the algorithm. They could always drop a dose for safety, absolutely, in that, but they couldn't change the dose that was selected or the randomizations or the decisions, but they could always drop a dose for safety. Two things happened, and they've made this public. The high dose, actually, the DSMB was thinking about dropping it because of the high heart rate, but the RAR had already zeroed it out. And so they were very happy with that, and they didn't have to drop it. But they said, we watched the algorithm and they never touched it, but said it did everything that we wanted it to do and expected it to do. So by walking them through ahead of time and describing all that, describing the rules, they absolutely followed it, and they actually came to very much appreciate the design.
Thanks for that, Scott and Michelle. I'll move on to the next question, which is from Mark McLean. Scott, would you like to take this, please? So the question is, when the trials are running, I assume the randomization is performed in a custom-built algorithm system. Typically, they are integrated with the EDC, or is randomization done offline?
I am going to pass this one to Michelle.
Sure. So that's a great question. So the randomization, there is a specific randomization system for response adaptive randomization. It's often custom, sometimes it's custom built depending on the complexity of the response adaptive randomization. Most systems just have to have the ability to be able to update the randomization at certain points during the trial. It's very common for IXRS, IBRS, different randomization systems to be based upon lists that are created ahead of time. And in the planning stage for implementing these adaptive trials, we work with the groups and we discuss with them that the lists just have to be updated at certain points, and they have to be able to take their system and put in new lists. And with adaptive designs, we want to make sure we have kind of this preparation phase as we're getting ready to conduct them, so we can kind of go through some of the pieces that we need to work with to make sure that we can conduct implement the response adaptive randomization. Now, that's if it's a list-based system. A lot of other systems have the ability to take the probabilities for each treatment arm and then randomize according to those probabilities, and then the group would just transfer probabilities. But there's lots of different ways and different ways we work with the systems that are in place, but we want to make sure we have that interaction ahead of time. But there's definitely possibilities, but just requires some flexibility among the systems and ability for groups to be able to consider maybe a different way of doing the randomization than having a single list base, which they may have had in the past for the entirety of the trial.
Excellent. Thanks for that, Michelle. The next question is also from Mark McLean. He's asking, does the frequency of adaptation of rando or enrichment only happen on predefined intervals, example, via the robots, or DSMB, or can it be truly dynamic?
Pre-specification is key. And Scott may want to add to this after I'm done, but the design is pre-specified in the protocol, and the pre-specification includes the timing of the interim analyses. Sometimes the timing may be based upon a trigger as far as a certain number of subjects randomized. Maybe an interim analysis happens at 100 subjects randomized or 200 subjects randomized. As soon as that trigger is hit, the interim analysis is conducted. Things have been prepared ahead of time that we're ready to go, and we conduct it. Sometimes interim analyses are conducted time-based, where they start at a certain point after a certain number of subjects have been randomized, and then additional randomizations, for example, in the Trulicity trial, happen every 2 weeks. But this is pre-specified, and it's not chosen as we're looking at the data or as the DSMB is looking at the data. The key is that the plan is specified ahead of time before data is seen, before unblinded data is seen.
Yeah, and that could be number of events, which comes somewhat dynamically, but it's well defined when it happens. So an interesting thing, Michelle, is we've run into now, one particular trial was supposed to do an interim at 200. And because of the coronavirus, patients can't come in anymore. And so now this adaptive trial is sitting at 195 patients and they're wondering what to do. So we do run into cases where weird things are happening external to trial, where we might institute an interim analysis 5 patients early because patients can't come in, and we may have a 3 or 6 month delay in the trial. So it's scientifically appropriate to do that. So that's a weird thing that might happen in the timing, but we would rather those not happen.
And I think to add on to that, one of the keys with that is that decision is made by the blinded sponsor team, and Scott being the blinded statistician advising. The DSMB is not weighing in and advising whether or not an interim analysis should be conducted because they've already seen the unblinded data. So they will get the decision from the blinded sponsor and the blinded trial team and the blinded statistician as far as what's being recommended, and then they move forward with it. They don't weigh in and decide what to do.
Sounds good. Thanks, Michelle. Mark has also commented, excellent, thanks for the great clear answers. And there's this question to you, Michelle, from Mami. Michelle, can you please talk through any recommendations or factors to consider when viewing unblinded data through interim analysis?
Sure. So when I'm on a DSMB or I'm on an SAC and I'm looking at the results of the interim analysis, I mean, the first thing I look through is just that it logically makes sense, that it looks like the data that I'm seeing, the summarization, it makes sense with the outcome of the results. The other thing that I'm looking at is just seeing the quality of the data to understand the missingness, and then to understand what the resulting decision was and how that was triggered. So I need to understand ahead of time what the possible adaptations were and the triggers for those adaptations. And then I'm looking at to make sure that really what was intended in the design is really what happened in the data I'm seeing, and whether there's anything that just doesn't make sense or doesn't logically make sense. And usually when that happens, I should say a skilled statistical analysis committee has really dug into the data and understands the data, and they're going to present the information in a way that if there are issues, they're going to put them out there to hopefully have that discussion with the DSMB and alert them to that. So it's very important for the statistical analysis committee to understand the data and understand the analyses they're running. Yes, this is all according to a pre-specified protocol, but it's not a push the button and don't look at it. You need to make sure you understand your data and understand the results and make sure it all makes sense.
Thanks for that, Michelle. This is the last question that we can take, unfortunately. We're running ahead of time. This one's from Leonid, and he is asking, either to Scott or Michelle, could you please comment on the process and information provided to DSMB in the situations of continuous response adaptations, that is, probabilities dynamically changing after individual patient outcomes are obtained?
So I'll take, you want to go, Scott, or do you want me to go?
I'm not exactly sure the question. Is this where an individual subject changes from one interim to another, their data change, or the outcome is continuous, Michelle?
I may have interpreted it differently. Can you read it one more time, Anisha?
I think we are, sure, I'll read it one more time. Could you please comment on the process and information provided to DSMB in the situations of continuous response adaptations, that is, probabilities dynamically changing after individual patient outcomes are obtained?
Okay, so sorry, I'll be really quick because I know you're out of time. So we create a report ahead of time. And those probabilities go into a report. It might be 10 to 15 pages that summarize the data. And the committee gets used to reading that report. The first couple times we walk them through detail by detail, and then they may get the report every Wednesday, for example, and there's no meeting, but they're looking at it and they're seeing this report that's meant for human eyes in order to digest the dynamic aspect of the trial.
Thanks for that, Scott. We do have an evaluation at the end of the webinar, which should show up after the webinar ends. Please take a moment to provide your valuable feedback. On behalf of the Australian Clinical Trial Alliance and our presenters from Berry Consultants, thank you for joining us today and have a great rest of your day.
Thank you. Bye.
Bye.
Thank you.